afternoon, everybody. This is our first session on the topic of psychosis for grand rounds for this year, and it's a pleasure to introduce Andreas Kruse, who is a fourth-year resident, and I've been working with him since his second year. He's in the research track, and he's going to present to us a summary of his paper that we published last year on glutamatergic dysfunction in schizophrenia. So I welcome everybody, and I hope that you enjoy his presentation, and then you have lots of questions for us. Go ahead, Andreas.
Well, thank you, Dr. Bustillo, and I want to also thank you for the opportunity for me to present at the Grand Rounds today, all your mentorship and support, both for the publication itself, our other research, and preparing me for the presentation today. So thank you very much. And we're going to start out by going over the learning objectives for today. So what I wish to convey here is to talk about glutamatergic dysfunction as a concept, as a theory underlying the psychopathology of schizophrenia. We're going to then move on talking about neuroimaging studies, spectroscopy studies, looking at glutamatergic activity throughout the course of illness in schizophrenia. And then we're going to move on and we're going to look at other domains of research for glutamatergic neurotransmission and signaling, talking about post-mortem studies, psychopharmacology, and genetic studies, and a brief mention also on measuring antibodies in serum. So let's get into it. So a bit of the background. So we know that glutamate is the main excitatory neurotransmitter in the human brain. And if glutamate signals unregulated, high concentrations has been associated with excitotoxicity and, in a more chronic stage, neuronal death. And therefore, physiologically, there's a system to regulate glutamatergic concentrations within tight control. So this image here is a representation of the glutamatergic synapse and the glutamatergic neuron. From a glutamatergic neuron, the neurotransmitter is released into the synaptic cleft. And when concentrations are high, there are specific reuptake transporters transporting glutamate into astrocytes, adjacent astrocytes. And from there, glutamate in its active form gets metabolites into glutamine. And we're going to come back to glutamine, but it gets synthesized into its inactive form, glutamine, and then it's transported back presynaptically into the neuron. And this serves to control the concentration within the synaptic cleft. Also worth mentioning, it's an energy-intensive mechanism. It is metabolized from this process, are closely linked to the Krebs cycle, and it constitutes a significant portion of the energy demands of our brain. So if you want to look at glutamate and investigate it in terms of imagery, how does that work? So that brings us to this imaging modality, this imaging technique called proton magnetic resonance spectroscopy, 1H-MRS. So it's a non-invasive technique, and it allows for measurement not just of glutamate, but a multitude of different molecules within the brain. So the hardware is the same as a regular MRI, magnetic resonance imaging. And how it works is that within a magnetic field, you're going to have protons oscillating and giving out a specific radio wave frequency. And because the proton composition will be different for different molecules, you're going to have a unique frequency for each type of molecule that you wish to investigate. And this data, obtaining these radio wave frequencies are then processed into different types of software, and the output typically looks something like this. So you have on the x-axis, you have what we call the chemical shift, which is corresponding to the unique signature of a specific molecule. And on the y-axis, you have corresponding amplitudes of such molecules. So in this example here, you see that the major peak here is for a metabolite called NAA, which makes sense because that is the most abundant neurometabolite in the human brain. And what you do is that the area under that amplitude is referenced to water within the same tissue, and that gives us an estimate of the concentration in the particular region that we're looking at. And you can also see that right next to the NAA peak, there's this bracket here that constitutes GLX. And that is a combined measure of glutamate and its inactive form, glutamine, as I was referring to in a couple of slides back. So when we're going to look at different studies, some of them are going to investigate glutamatergic signaling as part of this compound measure, while others are going to look at the specific smaller peaks of glutamate and glutamine separately. So this brings us to a model of illness, a model of schizophrenia patho-physiology. I am sure many of you are familiar with. This is representing, is an image representing a somewhat simplified mechanism of how this is happening. And on your left-hand side, well, actually on the right-hand side, you have a typical excitatory glutamatergic neuron, which from its synaptic cleft here expresses glutamate, signals with glutamate. And glutamate, one of its targets is the NMDAR receptor. And NMDAR receptors are typically found on inhibitory GABAergic neurons in the brain. And one theory is that if this receptor is not working properly, it doesn't signal at a consistent level to the inhibitory neurons, and they don't express GABA to the same extent that they typically do, which will constitute a reduction of GABA and a disinhibition of the excitatory neurons. So that's why we have this model of why would we see early in disease increased levels of glutamate when its receptor is actually dysfunctional. Wouldn't it make sense to be the other way around? But the reason for that is this mechanism of GABAergic disinhibition. So there has been studies looking at this and particularly concentrations of glutamate, glutamine in the prefrontal cortex. But what's worth mentioning, because here we're bringing a new concept, we're talking about ketamine. And ketamine as well as PCP. They are agents which for a long time clinically has been observed to elicit psychotic symptoms. But not only that, not only the positive symptoms of a disease like schizophrenia, but also in a chronic sense, it produces symptomatology which is similar to the negative symptoms and the cognitive symptoms that we see in schizophrenia. So what we see here on the left-hand side is a study where ketamine, sub-anesthetic ketamine, was acutely infused into healthy volunteers. And with the imaging modality I was mentioning before, levels of glutamine in this case were significantly elevated compared to saline. And another study the same year also looked at not only concentrations of glutamate, but also how these concentrations corresponded to positive symptoms of the PANSS score. And the higher concentrations in the prefrontal cortex of glutamate, the more pronounced the positive symptoms were, according to this scatter, represented by this scatter plot here. So let's talk about these studies, the imaging studies that has been done looking specifically at glutamatergic signaling. So we're going to talk about a couple of different types of studies. The large one are going to be cross-sectional studies, starting up with this meta-analysis. And then we're going to talk about a longitudinal study as well. So this is a spectroscopy study. It's around 8,000 subjects and 8,800 controls. And what they did, they looked not just at people with chronic schizophrenia. They had several subgroups constituting first episode psychosis, high-risk individuals, and treatment-resistant individuals. And they didn't just look at one brain region. They looked at, I think it was a total of 14 different brain regions and expression of glutamate. And these are sort of the highlights from this study. What they consistently found was that the combined glutamate-glutamine measure, the levels of GLX in the basal ganglia, were elevated for all the different stages of the illness. And then they also looked in the prefrontal cortex in an area called the MCC, medial cingulate cortex. And in there, they saw that among people with schizophrenia, both the combined measure and glutamate were increased. But if you removed patients who had a treatment-resistant condition, the expression was actually reduced instead of increased. And then another, this time a mega-analysis by Merritt et al. From a couple of years ago, looked at a slightly smaller sample, well, a smaller sample, but still quite significant. And in the medial frontal cortex, they also observed this correlation that glutamate and the combined measure were lowered in schizophrenia. And they didn't have a specific treatment-resistant subgroup, but the findings were consistent with the finding of Nakahara et al. I mentioned before, when they removed such subgroup. And what they also looked at, which is very interesting, is that they looked at antipsychotic dosing. And in the same region, the medial frontal cortex, for every 100 milligram of chlorpromazine equivalents, there was a 0.10 reduction of glutamate concentration. So that speaks to somewhat the neuroprotective properties in this region of antipsychotics. And now let's move on to longitudinal studies. So the one we're going to mention here is a really interesting study. Of course, it's going to be a smaller sample. And this group in Mexico, they looked at 24 antipsychotic naive patients with a first episode diagnosis, and they also did not have any history of substance use, comparing them to healthy volunteers. And at baseline, the first episode group had higher levels of glutamate in the striatum and the cerebellum. After these initial imaging, the patients got 4 weeks treatment with risperidone. And you could see that glutamate actually changed in the striatum, but it remained unchanged in the cerebellum. So this is interesting because we all know that striatum is very implicated in the pathophysiology of schizophrenia as a dopaminergic structure of the basal ganglia. So these findings in this study may be linked to the dopamine blocking effects of antipsychotics in the striatum, that it actually also modulates the expression concentration of glutamate in that brain area. And now let's talk a bit about, so we have the NMDAR hypofunction model, and what some groups have looked at is an immunologic mechanism for why the receptor might not be functional. And these couple of groups here look at first episode psychosis, and how many of these had a positive NMDAR receptor antibody. And we know this from clinical practice. It's a small minority of our patients actually will have a positive NMDAR receptor. A slightly larger study, well, a larger meta-analysis back in 2014 showed a prevalence rate of around 8% of anti-NMDAR receptors being positive. So we know that it's a minority of first episode or schizophrenia patients who have positivity to the NMDAR receptor. And we also know that some persons without psychosis will have a positive result on serology, but they will not show any signs of clinical signs of schizophrenia. So this study is also worthwhile mentioning. It's interesting because they looked at first episode psychosis, comparing it to healthy controls, and not only did they, so they already assessed that this subgroup had positive NMDAR receptors antibodies, but they also looked at clinical scores, PAN scores, and there was a positive correlation between levels of the antibodies and the severity of both positive and negative symptoms. So for this subgroups, mechanistically, it makes sense that an altered NMDAR receptor represents positive and negative symptomatology, but it's far from the only mechanism in which these symptoms occur. So that brings us into a different domain of research, post-mortem studies. So these are pathological and genetic studies of brain tissue for patients who have passed, patients with schizophrenia who have passed. In this domain, of research, you can look at structural changes in glutamatergic neurons. You can look at mRNA changes, and you can look at different types of protein expression, including receptors, transporters, enzymes, and agonists to the receptor. So this study here is a comprehensive review of such post-mortem studies. And they talk about how many studies have found reduced dendrite length, glutamatergic dendrite length, but also complexity of these glutamatergic nerve cells. And they also talk about this marker of the axon boutons of glutamatergic nerve cells called synaptophysin, which has been shown to be lowered in schizophrenia. And they also looked at specific areas in in the human cortex, the auditory cortex, and DLPFC, which showed reduced pyramidal volume in this specific layer. And this is another study. It's a review of 5 smaller studies, and they looked at mRNA and protein for a specific compound. It's called a subunit of the NMDAR receptor in the prefrontal cortex. And when they looked at effect size from these 5 studies, they found reduced expression of both NR1 mRNA as well as NR1 protein. They did look at other subunits too, and did not find the same correlation of reduced expression. So to summarize, mRNA and protein of the NR1 subunit for the NMDAR receptor are reduced in the prefrontal cortex. Glutamatergic neurons have been described to be reduced in size in the prefrontal cortex, both in terms of dendrites, axons, and cell bodies. So all these findings together are consistent with the NMDAR hypofunction model. But it's important to remember that a significant limitation of post-mortem studies is that you're not going to have any first episode psychosis subjects or high-risk subjects. These are going to constitute people who have had chronic illness and typically been treated for a long period of time with antipsychotics. So this brings us into the next domain, genetic studies. So we all know that schizophrenia has significant heritability, and there are several ways to investigate genetic risk factors and genetic expression in schizophrenia. We have what's called genome-wide associations, which not only look at high-risk genes and genes that are coding for specific proteins, but the genome as a whole, even non-coding regions. And these are called genome-wide association studies. And then you have other types of studies looking at more rare variants within the exon, the coding region of the DNA, and those are called whole exome examinations. And the last type of study, also conveying more rare variations, is called CNVs, and that's good to look at connectivity within a network of genes. So this is the largest genome-wide association study to date that we know of. It has about 75,000 schizophrenia patients, about 3 times as many controls, and they identified common variants at a total of 287 loci, constituting both inhibitory and excitatory cells. And it's interesting because they identified also several risk genes encoding proteins which modulates a glutamatergic receptor activity. And it was not just for the NMDAR receptor. They also found risk genes encoding ionotropic, so channel receptor of ions, and metabotropic receptors, abnormalities within metabotropic decoupled receptors within the glutamatergic signaling system. So this image here is a representation that these genes that were found all are on the postsynaptic component of the neuron, as opposed to the presynaptic component. Moving on to the whole exome examination study, and this is still a very large study, but comparatively smaller, 25,000 schizophrenia patients and almost 100,000 controls. And they detected group differences in 10 different genes. So this is going to be smaller because, again, these genes confer direct coding to proteins. So these are looking at specific genes apart from general genetic material, non-coding genetic material. And they found 2 genes which were related to glutamatergic function. So this is a different subunit of the NMDA-R receptor. And they also found a subunit of another glutamatergic receptor called the AMPA receptor. And what this image here is representing is the different localizations of the NMDA-R receptor subunit gene in cases compared to controls. So it's, I think it's 8 or 9 different variations among the cases, while only a couple for the controls. And lastly, talking about the CNV study, which is one of these were done a few years ago. And they had around 20,000 patients and 20,000 controls. And they identified 8 loci with deletions or increased risk of schizophrenia. And these were genes coding for synaptic cell adhesion and scaffolding proteins. But they found, in terms of glutamatergic findings, they found deletions and increased risk for 4 different receptors listed here. And what's interesting is that these receptors constitute a subnetwork where all these genes are connected. But in patients with schizophrenia, this subnetwork have more genetic losses than compared with healthy controls. Okay, so summarizing what we have learned from genetic studies. So there's been several large studies which have detected several different genes involved in glutamatergic neurotransmission. And what's really interesting is that although these are large studies, they identify many different risk genes. They have not described any risk genes involved specifically with dopaminergic neurotransmission. So despite this, glutamatergic genetic factors are only accounting for a small proportion of the overall heritability in schizophrenia. So there are many other genes and genetic burden that conveys risk. To the extent that the findings from genetic studies does not support a major role for genes known to impair the NMDA receptor. So moving forward, this is also a really interesting area of research, and this is the pharmacological studies that has been trying to target the NMDA-R hypofunction model, and not only to the receptor itself, but also different mechanisms targeting glutamatergic transmission. So this list is a representation of the different types of pharmacological targets and trials which has been developed. So we have co-agonists to the receptor. We have glycine, who is such a co-agonist, but it's a neural transporter being affected, and metabolizing enzymes of co-agonists. And we're going to talk in a bit more detail about allosteric modulators of both the AMPR receptor and metabotropic receptors. And then there's also has been some research in terms of NMDA-R antagonism, which makes sense to sort of reduce the dysfunctionality of the receptor and reduction of glutamatergic excitotoxic overload. So starting out with the co-agonists to the receptor, we have glycine and serine and D-cycloserine. So these were short-term trials described, mostly males, and what they tried to specifically target with this were negative symptoms. These studies were typically also additions to ongoing antipsychotic therapy. So like olanzapine or clozapine, these drugs were given adjunctively to antipsychotics. And there was no benefits for either of these three agents, but there was, for the combined glycine and D-serine study, there was some mild improvement noted, which is represented here. And then we were also, I was also talking briefly about the glycine transporters. So these agents, sarcosine and bitopertin, were both agents within this class. And there were initial studies with both of these agents which showed promising data. And this image here is representing the results of the bitopertin trials at 24 weeks, relatively large RCT of 600 patients, using it in addition to antipsychotics. And for neither dose compared to placebo in these larger trials, there was no significant effect. And the same for the other agents, sarcosine, I think it should be sarcosine, looking at positive, negative, or cognitive symptoms when used alone. Another agent is AMPAR modulators. Ampakine was such a pilot agent that was added onto clozapine. Initial data was positive. Larger follow-up study in the same group did not find any significant results. A subsequent meta-analysis a few years ago looked at this agent and other modulators of both NMDA and AMPA, specifically for cognition in schizophrenia, and there was no significant differences found on overall cognitive function. So this agent is really interesting. It's an agonist to a presynaptic metabotropic glutamatergic receptor. And when it's activated onto that receptor, it senses, it regulates the concentration in the synaptic cleft, in that when there are high glutamate concentrations, this receptor will ensure to reduce further glutamatergic release. So an initial study back in 2007 did show promising antipsychotic effect when comparing the agent to placebo and olanzapine. This caused funding for a larger follow-up study, which unfortunately did not show any changes in positive or negative symptoms. However, the same group looked more closely at their data. They did a reanalysis, which suggested that this agent might have some positive antipsychotic effects among schizophrenia patients very early in their illness, and if they've been unexposed to antipsychotic drugs. So we're going to talk a bit more about this later, but this makes this agent really interesting to look at in the setting of early illness, where we know that there's a population of patients which have increases of glutamate in the striatum. So to summarize, so there's been several large studies, large pharmacological studies of glutamatergic agents, none of which have resulted in any approved treatments. And there's been two strategies used, one looking at agonists at the glycine site of the NMDAR receptor, as an adjunct to antipsychotics for negative or cognitive symptoms. The other approach is looking at modulating or downregulating the glutamatergic release presynaptically through allosteric modulation, and these agents are typically used alone. And what's important to note in the sort of unfortunate lack of significant findings is that this is consistent with other non-dopaminergic approaches to pharmacologically treat schizophrenia. We don't have agents in other pharmacological domains either. And it also highlights how important it is to do further research and learn more about the underlying pathophysiology to be able to develop novel therapies. So these slides here, I strive to sort of summarize what this presentation and review is about and what we know so far. So what we do know, we know that people who without an underlying psychotic illness who are exposed to ketamine or PCP will experience both positive symptoms and longitudinally, chronically also negative and cognitive symptoms. And we know in controlled research settings that acute infusion of ketamine will increase cortical concentrations of glutamate on spectroscopy. And we also know that spectroscopy data has shown that glutamate is increased in the basal ganglia in the early courses of disease. And it seems that antipsychotic treatment moderates this increase of glutamate. And it's important to also note that despite the magnitude and the number of studies the spectroscopy studies which have been done, hasn't shown any association between the severity of symptoms and the different levels and changes of glutamatergic concentrations. And we also know that antibodies directed to the NMDR receptor seems to play a minor role in first episode patients and the illness overall. We know that post-mortem studies have shown several different microscopical changes in terms of neural cell volume, dendrite complexity, and we also know that the subunit, the N1 subunit of the NMDR receptor has been reduced for mRNA and protein in post-mortem studies. Genetic studies have detected small risk effects, both for common and more rare variants of schizophrenia. But we also know that the genetic burden seems more directly involved with glutamatergic systems than it does with dopaminergic systems. Pharmacological trials looking at agonism of the glycine site and as an add-on, looking at negative and cognitive symptoms versus allosteric modulators in monotherapy targeting psychosis. And none of these approaches have found consistent promising data. So what conclusions can we draw from all of this? So we know that glutamatergic abnormalities are present in schizophrenia. And some of these, especially the basal ganglia related findings, are related to the etiology of the disease. The prefrontal and the basal ganglia findings. And there's a few genetic loci that convey risk for glutamatergic transmission, but they have only a small part of the overall genetic risk. So looking at these increases that has been observed in the basal ganglia and the striatum, if glutamate is increased, we also know that dopamine plays a pivotal role. Like, are these increases actually related? And this is an area which still could support a lot of studies. The one study that we did find looking at this specific correlation did not support this. And it's also important to highlight that increases or reductions of glutamate or other forms of glutamatergic metabolism in other brain regions or other illness stages are more inconsistent and generally harder to interpret. So, as I mentioned in the beginning, we have the postulated model, the theory of NMDA hypofunction causing disinhibition of glutamatergic neurons and continuously increased levels and release of glutamate. And in a chronic stage longitudinally of the illness, these glutamatergic neurons might actually retract to reduce the risk of the toxicity that an overload of glutamatergic transmission produces. So that's why in chronic settings, you can actually see reductions of glutamate on spectroscopy data. And that would also be consistent with literature that has described overall cortical thinning in schizophrenia. So these last couple of slides, I'm just gonna mention and not go into detail explaining what these constitute, but this is interesting because it takes us into future prospects, future sort of promising venues to further our understanding of glutamatergic signaling and neurotransmission. So these are modalities within neuroimaging. So these are both invasive approaches where 13C-MRS is looking at glutamate and glutamine dynamically, not at two separate compounds, but rather representing the physiological reuptake and transportation back into the neuron I was talking about earlier on. And the other approach is using PET, and in which you have radionuclides binding to specific markers. And such markers could constitute both ionotropic and metabotropic NMDAR glutamate receptors. And both these approaches, they require longer acquisition times, more processing of data, really big imaging. So the areas of interest would be really large. And so there are logistical challenges to meaningfully practically implement these into research, but we might see these sort of develop and being implemented in the next few years. And then in the domain of post-mortem studies, we have something called brain transcriptomics, which is actually a GWAS, genome-wide molecular approach, looking at post-mortem tissue. And one sort of pilot study showed some synaptic downregulation a few years ago, and larger studies may actually reveal how this downregulation affects glutamatergic dysfunction. And I don't know if Dr. Wiley is here today, but he had a great presentation on his research on the research day a couple of days ago on circular RNAs. And this is interesting to look at glutamatergic signaling. So these are non-coding RNA. They are expressed overall in the brain and especially in the prefrontal cortex. And one such circular RNA called Homer1 has been found in the prefrontal cortex from patients with schizophrenia. So one avenue of research could be measuring circular RNA levels serologically in a high-risk group while comparing it to spectroscopy data in a time frame of conversion. That could serve to highlight the link between genetic risk and biological expression. So I think I'm getting towards the time point. Yes, that was the last slide. So I really appreciate the opportunity to be able to present to all of you today. And I will try my best to answer questions and also let Dr. Bustillo chime in for any questions that you guys might
All
have.
right. Well, thank you, Andreas. You finished right on time as we practiced earlier this week. So we have several minutes for questions. I want to encourage everybody. There's no silly questions. We understand that this is a very technical and complex area. So I want to encourage you, any questions, that doesn't mean we'll be able to answer them, but I do want to hope that you will feel comfortable asking. Anybody? So I want to sort of get us started, and so maybe Andreas, so maybe you can comment on what are the main findings in which of these domains or areas of research there's been findings for the dopaminergic abnormality in schizophrenia, and how do you contrast those with the areas in glutamatergic research where there have been findings? Do they overlap? Post-mortem in glutamate, post-mortem in dopamine, pharmacology in one or the other, genetics. Can you say something about that?
Yeah, I did comment on it specifically in terms of the striatal structure. I think there was a lot of expectations and hope that there would be more of a correlation there. And I think that would be maybe with these new techniques that are being developed and the sophistication of neuroimaging studies, that we can learn more when the voxels get smaller, the areas of interests get more sort of highly defined, that we could learn more from dopaminergic signaling in different separate basal ganglia structures, and contrasting that with glutamatergic signaling. That, as you know, that's one of the projects we are working on. And in terms of the pharmacological studies, that was I've been thinking about that when working on this quite a bit, how there can be sort of this gap between genetic risk not being easily conferred into the dopaminergic system, and yet that is where all our effective medications in terms of positive symptoms are. That's pretty much the effect in treatment of psychosis that we've seen so far. That's where it's allocated. So that is a really interesting gap to see why that discrepancy exists.
Yes, and so just to add to that, just as a take-home message for most of you, is that in terms of dopaminergic mechanism of psychosis, the strongest findings are the pharmacological studies. We know that D2 blockers work for psychosis, is the only thing we have, and the neuroimaging findings, which we know from PET literature that there's increased release of dopamine in the striatum in psychosis. Yeah, those are the strongest findings. For the glutamatergic model, the strongest findings, paradoxically, are not the neuroimaging and certainly not the pharmacological, but the genetics findings that Andreas summarized so nicely, and the post-mortem findings. The post-mortem and genetic findings for the dopaminergic models are basically non-existent. There's no findings. So that's something that is very it's curious, and it's something that we just need to keep in mind. Any other questions from anybody?
Hey, Andreas, this is Will here.
Hey, well,
Thank you for your talk. I really enjoyed listening to it, and thanks also for the quick shout out. I didn't have a question, but just a quick comment. I appreciated your summary of the involvement with astrocytes in regulating glutamate metabolism. It reminded me of some of the studies I've been reading about circular RNAs, looking at their export via exosomes within the neuropil, and so just kind of imagining them as one of the part of the crosstalk between neurons and astrocytes and other parts of the brain parenchyma as part of this interconnected regulatory network. So yeah, I just thought that was kind of a cool thing. It related to the bit you had on astrocytes, and thanks again.
yeah, absolutely. Thank you, Dr. Wiley. I'm not sure if you were familiar with the type of circular RNA that was mentioned here, and I know it's, maybe Dr. Bustillo can fill in on this. I know it's innately linked to glutamatergic signaling, but the specific mechanism escapes me now.
Oh yeah, Dr. Wiley knows about Homer 1. You want to
share that's
the
actually
relationship between Homer 1 and glutamate, Will?
I'm not sure about specifics with glutamate, but that paper is actually out of the Melios lab, who's one of my research mentors in the neuroscience building. Yeah, they did a lot of work with learning reversal, finding that Homer 1 was required for neuroplasticity in learning reversal in a mouse model.
All right. Anybody has any other questions before we let Dr. Kruse go?
Yeah, I have a question. This is Robbie DeBurler. I'm a PGY1. I'm watching my sick baby today, so if you hear him in the background, I apologize. I was curious. I saw some research a while back when I was looking into something called computational psychiatry, and it's kind of stuck in my mind. They did a model of a inhibitory interneuron, which I guess has reduced concentration of NMDAR receptors in people with schizophrenia. And I'm curious, with your model of like the relationship between glutamatergic and GABAergic neurons, is one of those an interneuron, or is that, am I making that connection correctly?
Yes. Yeah, so I'm not sure if I mentioned it, but the GABAergic neuron.
You want to pull the slide?
Yeah, I was trying, but it doesn't want me to. Here we go.
That's it.
So it doesn't say here, but you're right that the GABAergic neurons act indeed as what we call interneurons, and that's the terminology used for and the sort of, as is the case in many regulatory systems, it's important to control the sort of evenly distributed signaling from the glutamate to then get a feedback loop to ensure that the signaling doesn't get disinhibited. So the receptor itself is on these interneurons, the GABAergic interneurons.
Yeah, thanks.
I don't know if you want to answer that, Dr. Bustillo.
Yeah, I do. So the next thing they mentioned was that in people with schizophrenia, their working memory is kind of compromised, and that they can't distinguish between stimuli as well, and that was related to the lack of inhibitory control of related stimuli to a certain. So they can't distinguish between different stimuli because the non-relevant stimuli aren't being inhibited. And that's kind of what they base their computer model of this neuronal system on, and that you have similar effects when you give someone ketamine. So is that kind of consistent with everything that you were teaching us today? I just want to.
I actually haven't heard about that specific.
Yeah, I can add something. So I presented on Wednesday from another study that we have, a pilot study looking something very similar to the task that you're mentioning, Robbie.
Mm-hmm.
So the difficulty in distinguishing two very similar auditory stimuli.
Right.
I mean, and this is what the classic paradigm is, the oddball paradigm. And patients with psychosis are impaired in this task, and there's no question that they are impaired in this task, it's hypothesized that is related to NMDA hypofunction in GABAergic interneurons, but there's no direct evidence of that. It's very hard to measure GABA in the brain, and the way we measure it right now is in very big chunks of brain, like 30 or 40 cubic centimeters. So we don't have the resolution to be able to say where we're looking anywhere close to a layer where there's mostly interneurons. So it's mostly just a theory at this point. Okay, I think we're done with time, so thank you very much. And again, thank you to Dr. Kruse for valiantly and gallantly coming forward and presenting grand rounds as a fourth-year resident, and we hope that he continues to do good work like this one. Have a good weekend, everybody.
Thank you.